A very warm morning to one and all present here. I am CS Tanmayaa, a research associate this YSP working under my mentor, Dr. Siddharth Pandey, on the project title named Identification and Characterization of Terrestrial Analog Sites in India to Support Moon and Mars missions. Now, before I move forward with my presentation, I wanted to ask you guys a question. We're all sci-fi nerds here, I'm assuming. So, when you watch the movie The Martian, have you ever wondered, like, if they had simulated the entire thing on Earth, with the right protocols, they could have probably, avoided the catastrophe, right? And thinking about that, you might be wondering what my talk has to do like with this lake in the background, right? So that's what my presentation is going to be about. It's going to be about the relevance of SOCAR. It is a high altitude salt lake in Ladakh, India, and how it is relevant as a Martian analog and its astrobiological insights. So before I move on with my work, I think it's only fair that I mention a big news that has happened, which is the setup and inauguration of a research base at the Soka Valley called the Himalayan Outpost for Planetary Exploration, or HOPE. So my mentor at Dr. Siddharth Pandya actually worked on this and this was set up in collaboration with ISRO, that is the Indian Space Research Organization, as well as the Mars Society Australia and the Mars Society. And I am particularly super excited about such a project because it's such a privilege to have a analog center in my own country, and I hope to contribute more to this field. So a quick note about the Hope outpost. There were two expeditions that were running from July 18th to August 10th, which were designed to mimic life support, isolation, and operational conditions that astronauts would face on Mars or Moon. So ISRO is planning to send manned missions to Moon first and then to Mars through under the Gaganyaan program, and to simulate those conditions to see how the low pressure or high UV exposure affects astronaut, their psychological state. All that is going to be observed at this base site. So the 10-day mission objective is as follows: is this to study critical physiological, psychological, and operational challenges, and to improve mission planning and preparedness for human space exploration in manned missions, right? And where was this set up? Like I said, it is set up at the Scar Valley. So, Sokar is a high altitude alkaline region in Ladakh, which is in the union territory of India. It's a very regional experience with to work to send humans out to verify the skills. It is because a higher visual experience to verify the number of spirit function, which project not minute, but they are pretty close to Martian conditions when compared to other sites on Earth. And then secondly, their geochemistry and sedimentation patterns. All the sediments found on Soka are mainly carbonate and there's alluvial sediments having boron content, which has also been observed on Mars. So this can be linked to prebiotic chemistry as an early life formation, and can explain the patterns of sediments formed on Mars. So if we study this site, we get insights about what has happened on Mars. Um, Thirdly, the permafrost and seasonal changes similar to early Mars. So since this region is drier, as in when compared to most places on Earth, um, and it does undergo some diurnal seasonal changes. This is very similar to the early Martian phase, the Noachian phase, epoch. Not sure if I'm pronouncing that right, but yes. So these seasonal patterns, they uh, 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 are parallel to what we have hypothesized how seasonal variation happened on Mars. So, like I said, so if we study this, we get insights on that. And lastly, preservation of organic matter. Because of the permafrost, because of the sedimentation and the permanent ice structures. It is a really good site for preservation of organic matters like lipid biofilms and other macrofossils. And so, if we learn how to look for those biosignatures on SOCAR, it gives us an insight on how to build tools and instruments and what laboratory techniques to use on Mars. So these are some of the other sites, other analog sites on Earth, which are similar to SOCAR in some aspects, not all, and how they are similar to Mars. Again, analog sites, they have various like types to it. Like analog is in their similarity with respect to geomorphology or geochemistry or biogeochemistry, climatology. So these analog sites are almost always in complete analog sites. So they don't mimic all of the conditions that are found in Moon or Mars. So this is one thing that I found pretty interesting while I was doing my review work, is that some of these structures, again, these are not, this is just for visualization, and I found it really interesting, is that there are the left side, A and C, are the Google images of SOCAR, and the right side, B and D, are the images that we have gotten from Mars. And as you can see, these physical structures are pretty similar. And if from the spectrophotometry analysis, they do have some similar mineral patterns and everything. So that's like even visually, SOCAR is pretty similar to Mars and these early Martian structures. So this is a rock glacier in SOCAR versus the Gale crater, and you have the solifluction lobes, so which are basically like alluvial deposits, and these are the Bosporus planum in March. So as you can see, the river, the dried up river channel and the silt deposit along the water shore. So why to study about SOCAR from an astrobiological perspective? So astrobiology is the study of life outside of Earth, like in space. It talks about the origin of life, and it talks about, how life can evolve or what, clues it can leave behind for us to look into. So the presence of extremophiles, that is life's adaptation to these particular environments. Preservation, like I said before, SOCAR's climate gives a perfect condition to have lipids and other microbial mats preserved in its ice sheets. And testing, that is to test our instruments, rovers, and everything on the site before we move on to the actual, planetary exploration. So extremophiles, just a quick intro to it, are organisms that are able to live in extreme conditions. So they have particular different genes and mutations that are not found in mesophilic organisms. And they wide, range of uses for humans, from an industrial point of view. And as you can see, these are the three predominant extremophiles found in SOCAR: alkaliphiles that are able to survive in basic conditions, halophiles able to survive in high salt concentration, and cyclophiles able to survive in low pressure and cold conditions. And their relevance to astrobiology, like I said, what clues they leave behind, what external products, biomolecules they leave behind, how they are preserved, how they are fossilized. All this we can see what role they play in the ecology of the environment. So this is a quick data that I've put together about the life forms present in SOCAR. So about most of it is archaeal life forms, and only like 27% of it is bacteria. And these are the major phylum of bacteria found on SOCAR. And what this means is that the general metabolic pathways of these genera found relate to carbon metabolism, amino acid biosynthesis, and methane metabolism. What this means is that they utilize the carbon and the methane present in the atmosphere, and they biotransform it to feed the food web and the ecosystem there. So for particularly the proteobacteria found in the soil of SOCAR, has the highest relative abundance, and it's known for its photoautotrophy, that is, making food from light, carbon dioxide fixation, and chemoautotrophy. Chemoautotrophy, as in getting your energy from, the ions, as in from the chemical components of biological organic matter. And yeah, these are the functional analysis, as in like most of the organisms are adapted to the cold environment. Some of them are adapted to the alkalic nature of sugar, and then these are the rest of the the functional expressions that these bacteria showcase. So yeah, so why we need to study these? And so we talked about that. Now, what is the future scope in this field? Is that it's a more metagenomic analysis for database formation. I worked on a limited data set and I found it pretty, it was a bit inconvenient. And if we do this, as in if we do proper metagenomic analysis, as in we collect samples and we see what different species are present, that can lead us to a search of useful extremophiles, as in more bacteria that we can use in our industrial products. Something that I want to give attention to is the immunogenicity or the pathogenicity of these organisms. So, most extremophiles are non-pathogenic, yes, but they are biofilm forming organisms. Biofilms are nothing but, it's like a layer that these bacteria form to protect themselves from environmental stresses. Now, most of the biofilms are, they do cause some sort of allergic reaction or infection to humans. So I was wondering if these extremophiles, rather than mesophilic bacteria, also cause those similar symptoms in humans. There aren't a lot of reported cases, but I feel like this is a new avenue that we should definitely look into, especially now that we're sending human scientists on these analog areas. And one more way that even if these are non-pathogenic, they can become pathogenic or virulent to humans through something called HGT, horizontal gene transfer. That is, they can uptake DNA from other bacteria, whatever bacteria I am carrying. Say I go to a site, whatever bacteria I am carrying on me, that those bacteria might transfer their virulent genes to these extremophiles and they can thus become pathogenic. So if we study about that, we can develop, vaccines or antibiotics that fight against this. So that is it. This was the work that I did for my young scientist program, and special mention to my mentor, Dr. Siddharth Pandey, and my teammates, Aaron, Shayla, and Lisbeth. I'm really, really thankful for Blue Marble Space Institute of Science to give me such an opportunity. And I'll always cherish this and work towards my research and scientific communication. Thanks a lot.
Great job, Tanmaya. That's awesome. I think we'll allow for one question with the time we have. Sanjoy has his hand up immediately. So I'll allow Sanjoy to ask the first question here today.
Tanmaya, thank you very much for your presentation. Really, really interesting. So as you know, the geology of Ladakh is very different from the geology of Mars, and India has other locations that are also good analogs to Martian environment. I'm thinking of Lonar Lake. Do you know if there has been some comparison between the biology that's found in at Sokar, which is more alkaline, versus the biology that has been found at Lonar, and if they're different and why?
The Lunar is an analog site in India, as you said. And I haven't particularly looked into it, but I am assuming the lake, as in the lunar crater, is either like neutral to a bit acidic environment. So the extremophiles found there are going to be different. But I do feel that there's not going to be much like too extreme of a difference, because the phylums that I showed, the Proteobacteria, Actinobacteria, they are all found in all types of soils. So there's not going to be, drastic differences, but obviously there are going to be differences because lunar crater isn't high altitude. It is like 400 to 500 meters above sea level, whereas Sokar is like 4,500-ish meters above sea level with higher UV exposure. and colder, way colder temperatures. So that in itself, has, basic, climatic, physical differences. So yes, the extremophiles found will be different. I personally haven't looked into it, but yeah, I hope that answers your question.